Hi everyone, this is Dr. Hassi. Welcome back to the channel. Today we are going to cover the study guide for BSC1010L, Module 3. So we have mitosis, meiosis here, Mendelian genetics, and chi-square analysis are the main topics that we are going to cover today. Okay, so we start with mitosis and meiosis cell cycle. Okay, so make sure that when you start studying for this module, so get down basic vocabulary like offspring, parents, inheritance, chromosomes, meiosis, fertilization, diploid, haploid, spores, gametes, gametophyte, sporophyte, and genetic variation. Just some examples for you. Okay, so let's start with mitosis first. Mitosis is division of nucleus. Basically, in the mitosis, we see actually in major regular cells happening in the part of the cell division. So during the mitosis, it starts with interphase. Basically, at the beginning of mitosis, start with the interphase. Interphase, we cannot see any chromosomes, but we know that actually they are duplicating and DNA and then making duplications. So when we see them first time, it's really prophase and they are already duplicated. You can see here in the mitosis. So in metaphase, they line up in the middle. In anaphase, they actually break up and they go in two opposite directions. And then telophase, they already move to form two separate nucleuses on the single cell, look like an H-shaped cell. So that ends mitosis. The next stage is obviously cytokinesis, where the cytoplasm actually cuts off and divides, and so a single cell becomes two cells. So what's so unique about mitosis is basically copies the cell and make two copies of exact identical cells. So everything is identical to the parent, single parent cell. In meiosis, on the other side, there are 10 stages. It's like two mitosis back to back. So most these stages actually duplicate, but because there is only one single interphase, they actually end up with halving the chromosome number at the end of meiosis. So even though we have four daughter cells at the end, but they are actually haploid chromosome number in meiosis. So it's a special cell division, happens in gamete cells only, and it produces egg or sperm cells. So that's meiosis. So make sure that you know all the stages of meiosis and you know what comes before, what comes after. And so remember, meiosis produces unique daughter cells, not identical. They are unique, unlike mitosis. There are also life cycles that, as you know, in every life cycle, meiosis and fertilization are the two big processes of the life cycle. So this can connect, we can connect this easily to Mendelian Genetics, which is the next lab, and chi-square, right? So Mendelian Genetics has a number of vocabulary, starting with actually allele, gene, different form of a gene, crossing over meiosis. Again, we just talked about diploid, haploid, dominant, recessive, fertilization, gamete, recombination, genetics, genotype, phenotype, haploid, diploid, heredity, tetraploid, triploid, heterozygous, homozygous, and hybrid. So these are all terms that actually start with Greg Mendel, which is considered the father of genetics, in his work and publication in the 1850s on pea plants. So as a model system. So in his findings, we know that basically if a genetic situation actually governed by one single dominant gene, it shows 3:1 distribution in F2. If it is governed by two dominant genes, its actually distribution goes up to 9:3:3:1 and goes on like this. If it is several genes, then it's not, no, we call them QTLs, and we can actually locate their locus on chromosomes and then map them on chromosomes, again, these QTLs or group of genes. So we know that genes are located in specific locations on chromosomes. And alleles are the different versions. They could be dominant or recessive alleles. In genetics, there are also like some other forms of dominance, like incomplete dominance, like we see in some plants, that they cannot completely dominate the other ones, or co-dominance, like we see in some blood types. They actually show they are both so strong, they show up together in the next generation. And there are also multiple alleles and test cross. Make sure that you cover all these, and epistasis, like if there are two genes, especially in animal systems, we might see epistasis happening. Okay, so these are all part of this chapter. Make sure that you study really this chapter very well and check the chi-square analysis. So if you have an unknown genotype, and if you want to find out what type of ratio that could be applied to this, you can come up with an hypothesis. Okay, this is 3:1 ratio, and then you can test your hypothesis using chi-square test. And then if you accept the hypothesis at the end, comparing the table value, you basically, so you found the ratio and then how many genes actually, how many genes involved in this genetic situation. So keep studying. Thanks for watching, and good luck with your exam and review. Please feel free to connect and subscribe to this channel, and see you next time.